we have discussed the reversible and irreversible cell injury, and also we have discussed the cellular injury and cellular adaptations types. Okay, so apoptosis, we are having three pathways in which the apoptosis occur, and these are the intrinsic pathway, the extrinsic pathway, and the perforin or granzyme B pathway. The intrinsic pathway is also known as mitochondrial pathway. The extrinsic pathway is also known as death receptor pathway, and the perforin gran enzyme B pathway. These are the three pathways. Okay, so apoptosis is a ATP dependent program cell death. Whenever apoptosis occur, it requires ATP. Intrinsic, extrinsic, and perforin granzyme B pathways. It activates caspases, that is cytosolic proteases, which leads to the cellular breakdown including cell shrinkage, chromatin condensation, membrane blebbing, and formation of apoptotic bodies, which are then phagocytosed. It is characterized by deeply eosinophilic cytoplasm and basophilic nuclease, and uh, pycnosis and karyorrhexis. Cell membranes typically remains intact with it without significant inflammation, unlike necrosis. DNA laddering fragments in multiple of 180 bp is a sensitive indicator of apoptosis. Okay, what is the indicator for apoptosis? It is DNA laddering, which is fragments in multiple of 180 bp. So how apoptosis occur? It occurs in three ways: intrinsic, extrinsic, and granzyme B pathways. And how it occurs by activating of caspases, that is a chain which is cytosolic proteases, in which cellular breakdown occurs, which leads to the cell shrinkage. First the cell shrinkage, then the chromatin material inside the cells condensed, and the membranes blebbing occur. This is the membrane and this is the blebbing. So blebbing occur, the chromatin material shrinkage occur, and condensation occur, and the cell shrinkage occur. It leads to the formation of apoptotic bodies, which are then phagocytosed, means which is eaten. Um, it is characterized by deeply eosinophilic cytoplasm and basophilic nucleus. When the cytoplasm is eosinophilic and the nucleus is basophilic, that is apoptosis. It leads to karyorrhexis, and the cell membrane typically remains intact without significant inflammation. That is, the cell membrane is fine. It is not like necrosis. The DNA laddering occur, that is fragments occur for multiple fragments in 180 BP appear, and it is a sensitive indicator of apoptosis. Now, what is the intrinsic pathway? Involved in tissue remodeling in embryogenesis, which occurs when a regulating factor is withdrawn from a proliferating cell population, that is, increased interleukin 2 after a completed immunologic reaction. Apoptosis of proliferating effector cells also occurs after exposure to injured stimulus, for example, radiation, toxins, and hypoxia. Now it is regulated by BCL2 family of the proteins, that is BEX and BEC are pro-apoptotic, which is bad for survival, while BCL2 and BCLXL are anti-apoptotic. Be clever life. This from this it can be remembered. BEX and BEC form pores in the mitochondrial membrane, release of cytochrome C from inner mitochondrial membrane into the cytoplasm, and then the activation of caspases occur. BCL2 keeps the mitochondrial membrane impermeable, thereby preventing cytochrome C release. BCL2 overexpression, for example, follicular lymphoma, decreased caspase activation, and tumorigenesis. This is the intrinsic pathway. Now, what is the extrinsic death pathway? It is a ligand receptor interactions. Fast L binding to FAS CD95 or TNF A binding to its receptors, and fast FASL interaction is necessary in thymic medullary negative selection. Autoimmune lymphoproliferative syndrome caused by the defective fast FSLL interaction, failure of clonal deletion, and increased number of self-reacting lymphocytes, presents with lymphadenopathy, hepatosplenomegaly, and autoimmune cytopenias. Now the perforin or granzyme B pathway, release of granules, that is containing perforin and granzyme B by immune cells, that is cytotoxic T cell and natural killer cells. Perforin forms a pore for granzyme B to enter the target cell. Okay, this is the intrinsic pathway, this is the extrinsic pathway, and this is the perforin granzyme B pathway. So in the intrinsic pathway, the DNA damage by radiation or ROS or toxins, misfolded proteins and hypoxia occur. It leads to the P53 activation, that is bugs and back, which leads to the BCL2, um, which execute Caspases, that is chain, and cytoskeletal dispersion occur. As a result, cytoplasmic bulb form. Uh, Now, in extrinsic pathway, tumor necrosis factor A, TNFR, and FASL and FAS, these are released, and it leads to the executioner caspases and leads to cytoskeletal dispersion. In Granzyme B, it leads to the executioner caspases. It leads to nuclear fragmentation, and as a result, ligands for macrophage cells happen here and apoptotic body, and it leads to the macrophages.